Anyway, I just want to welcome you all here. This will be the first Laura Randall Schweppe Endowed Lecture Series in Marine Science that we're having this year, but we have several additional ones planned during the year, so this is going to be a great series. I think the rest of them will mostly be on Fridays. Yeah, so anyway, so I'd just like to acknowledge this is through a generous donation in 1998, Dr. Irving Schweppe Jr. and his daughters Ann Ashman and Jane Scott endowed the Laura Randall Schweppe Endowed Lecture Series in Marine Science at the University of Texas Marine Science Institute. So this series honors the memory of a wife and mother who had a lifelong love of the marine environment, especially here in Port Aransas. The lecture series brings accomplished scientists to the Marine Science Institute to interact with its faculty, staff, and students, and especially to foster new ideas and research collaborations. So this has been an extremely successful series in the past. We've made all sorts of great contacts and new colleagues through this seminar series. So again, Anne, thank you so much for what your family has done for the Marine Science Institute. And Sharon Herska has been our seminar series chair this year, and she's just done a fantastic job. So I'm going to let her introduce our seminar speaker.
Thank you. Welcome, everyone. It's really my pleasure to present Rainer Amon. He is currently professor in the Department of Marine and Coastal Environmental Science in the College of Marine Sciences and Maritime Studies and in the Department of Oceanography at Texas A&M University. He received his MS in marine biology from the University of Vienna in Austria. He is Austrian. And his Ph.D. from the Marine Science Institute here in Port Aransas back in the day. And after he finished his Ph.D., he went to work as a research scientist at the Alfred Wegener Institute for Polar and Marine Research in Bremerhaven in Germany before beginning his tenure at Texas A&M University. His research has focused on the Arctic region for the last 30 years, generally working on carbon biogeochemistry in the Arctic Ocean and large Siberian watersheds. Over the last 10 years, he was also part of a large international project in the Gulf of Mexico to study deep water exchanges with the North Atlantic, a project in which we collaborated. Dr. Amon has extensive experience with international research collaborations, particularly with Russian, European, and Mexican colleagues, during which he organized and participated in numerous field campaigns, including a 2024 expedition to the Central Arctic Ocean. Before returning to the university duties in July of 2024, Dr. Amon served for 3 years as program director in the Office of Polar Programs for the U.S. National Science Foundation.
Thank you, Sharon, and thank you, everybody, for having me. Can you hear me okay? So today, I'm going to take you on a little trip from the Arctic, starting in the Siberian watersheds, going into the Central Arctic Ocean, the Nordic Seas, and then make a wild connection to the Gulf of Mexico. So bear with me. I have a lot of pictures, so maybe there is some entertainment involved in this. As I said, there will be a few different components to this presentation. The first one will deal mostly with river discharge into the Arctic. I was involved in the early phase of the so-called Partners program, which investigated the discharge of the largest Arctic rivers. That started in 2002. The second part of the talk will deal with the Central Arctic Ocean and how it connects to the Atlantic. And then towards the end, as I said, I will come back to the Gulf of Mexico through a deep water connection between the Arctic and the Gulf. The picture that you see here, in case you guys are wondering, this is a tributary to the Yenisei River in Siberia. So it can be quite beautiful and actually pretty warm. This gives you an overview of the Arctic region. You see immediately there's a lot of rivers flowing into the Arctic Ocean, and about 10% of the global river discharge goes into the Arctic, despite the fact that the Arctic only represents 1% of the global ocean volume. So a lot of river water that has a big influence on the oceanography in that basin as well. Along with the rivers, you see some white arrows, which shows you the surface, the general surface circulation of the Arctic, and some yellowish arrows, which represent the circulation of the Atlantic layer. So the Atlantic basically comes in here as an extension of the Gulf Stream, if you want, goes into the Arctic Ocean and basically flushes the entire basin at mid-depth levels. The general surface circulation goes from the west to the east and then turns around somewhere around the Laptev Sea area here and forms the transpolar drift, which is a very significant oceanographic feature in the Arctic Ocean. And then that surface water exits the Arctic towards the North Atlantic again. In terms of the river discharge, one of the unique things in Arctic rivers, as you can imagine, there's a lot of snow. So the discharge is very seasonal. And so on the top panel here, you see the flux of organic carbon or dissolved organic carbon between 2004 and 2021. And each of those spikes that you see here is basically the snowmelt signal. So somewhere between the end of May and early June, that snowmelt pushes out into the Arctic, carrying a lot of carbon with it. The contribution of this carbon into the Arctic comes mostly from the Siberian rivers. The North American rivers from Alaska and Canada are in yellow here, and you see that most of that signal comes from Eurasia. I'm not going to have too many of those crazy slides. I just wanted to point out that what you see here in blue is the actual water discharge from the different rivers that are listed here. So those are the 6 largest rivers that discharge into the Arctic Ocean. The Lena, the Kolyma, the Yenisei, and the Ob are in Siberia. The Yukon is in Alaska, and the Mackenzie in Canada, right? The snowmelt signal not only pushes more water into the Arctic, it also carries higher amounts of carbon. So what you see here in those dots is the actual concentration of a chemical that is only produced by land plants. And so it's a good tracer for us to track terrigenous organic matter anywhere in the ocean. And you see that during that snowmelt, you have a lot higher concentration of that organic carbon coming in. So it's a combination of the water discharge and the amount of carbon that is pushed into the Arctic during the late May, early June season. If you want to take a closer look, you see that all those rivers are not the same. And I would point out the Mackenzie River that has a lot lower concentrations of organic carbon compared to the other rivers. Early on in our work, we realized that something must be going on in the watersheds because the concentrations of carbon that we find at those downstream sampling sites were much lower than what we saw in the tributaries. And so the story that I have today is trying to think what it means to lose so much carbon, and if we are really accounting for that carbon that we are losing in the watershed. That loss is also reflected in the chemical composition of organic matter. And so all of you know sugars, all of you know proteins. Those are some of the components that we can analyze. In general, dissolved organic matter that's in nature, like in the ocean or even in rivers, we really can't analyze most of it. Sugars, amino acids, lipids maybe make up 5% of the total, but they can tell us a story. What sugars and amino acids tell us is that fresh material and material that is easily degraded by bacteria has a lot more sugar and a lot more amino acids or proteins. And so when you look at that, broken down in the different phases of that river discharge, you see that the snowmelt signal, what is here called high flow, always has the highest concentration of sugars and amino acids. And that means that that material is also easily degraded and might explain some of the losses that you see when you compare those four rivers to the Mackenzie. Now, why would the Mackenzie have lower concentration of that labile organic carbon? The Mackenzie has a lot of lakes, and so it takes the water a lot longer to go from the snowmelt all the way to the Arctic. And that additional time that the bacteria have to break down that organic carbon is reflected in lower concentrations of sugars and amino acids. So what is the long-term perspective? Again, going back to the Partners Project, what is now the Arctic Great River Project, we have now a 20-year time series to look at the cumulative discharge that is coming out of those rivers. And there were some surprises in there. You know, if you're familiar with the whole discussion of permafrost or other carbon storage in the high Arctic watersheds, there is a lot of carbon that's bound in the permanently frozen ground. If that ground is melting and that carbon is degraded, you are talking about a very substantial source of CO2. That's why people are thinking about those. And to the surprise of many people, the amount of carbon coming out of those rivers hasn't changed over the last 20 years. However, alkalinity and inorganic carbon did increase quite substantially over that time, while an important nutrient like nitrate actually has decreased. But for today, we want to focus on the DOC and how it might play into the story of increasing the inorganic carbon. Very, profoundly speaking, organic matter is respired, right? All of us, we eat food and we breathe out CO2, right? Bacteria in the environment do the same thing. They eat, if you want, organic carbon, and they breathe out CO2. And so that is one of the connections that you keep in mind. So breaking down organic carbon produces carbon dioxide, right? Very simple speaking. So what does Siberia look like? Just to give you an idea, this is a picture taken in June last year, not by myself, obviously, but by my collaborator. And it shows you the kind of water that comes off those Siberian watersheds when the snow melts. And so think about it like a tea bag, right? I often call it the Siberian tea bag. So if you have all that area covered in snow, sitting on all that organic matter, which I call tea, when that snow melts, it produces this dark brown colored water that flows off into larger rivers and larger rivers and larger rivers, eventually entering the Arctic Ocean. This is a picture of the study area and just a few pointers here. Why would we choose the Yenisei River? The Yenisei lies right here. And you see that there's a big difference between what is west of the Yenisei River and what is east of the Yenisei River. West of the Yenisei River, you have the West Siberian lowlands that are covered in peat bogs, and it's a very different ecosystem than the Central Siberian Plateau. But the Yenisei is so interesting because you get two different scenarios. You get the western scenario, which is very similar to the Ob River, which is another massive river in Siberia, while the eastern part, those are the typical large forests that you see in Siberia, is very similar to the Lena River. So by studying one river, you actually get information of 3 rivers that cover pretty much the entire Siberian area. So what we did then after planning this for several years, international collaborations are not always easy, especially if you go into areas that are extremely remote and things can go wrong. So you need to rely on the people you're working with, and that is a decision that takes a long time to make. You need to really know those people, which is why I visited Siberia, I think 3 times before we actually engaged in research. But after we did, we decided to do a proof of concept expedition, taking a ship from Krasnoyarsk, is where the laboratory of my collaborators is, and go down the river to approximately Norilsk, or at about 69 degrees north. And we were planning to sample that river while we are moving on it and to measure the flux of CO2 and methane coming out of the water. From the partners program that I introduced before, we already knew how much dissolved organic carbon is in the water, but we were suspecting that a lot of the carbon gets degraded in the river. And if it's getting degraded, it most likely goes directly into the atmosphere. And so we wanted to capture that. And so after visiting all the stations, we came up with that plan. And just as a side aspect here, so you understand the magnitude of changes during freshwater or during snowmelt. This picture was taken in July 2015, and you see the water level here is down there. If we would have been there a month earlier during snowmelt, the water level would have been up here. So this is basically the amplitude that the water level changes when the snow comes, the snow melt happens during that time. So a lot of changes. So this is the platform that we used. It's an old patrol boat from the Soviet era, and it's not a research vessel, obviously. And so in order to make measurements on a platform like that, you need to make a lot of adjustments. And some of the adjustments are shown here. So this would be your wet lab situation, where you basically work on deck and hope that the rain is not going to be too hard. But we also had a lot of instruments that needed to be installed. And so we basically asked the captain if it's okay if we weld some stuff onto his ship. Luckily, they don't mind usually. And so what you see here is a very simple structure of some metal that we found in a scrapyard. And that's really what we did. We went to a scrapyard and grabbed everything that seemed somehow useful. And this one actually carries an ADCP. So it's a device that allows you to measure current in the water. And the cable then, it's hard to see in this case, went into the kitchen, which was also our lab. So it's very tight spaces, and you need to find your way around it. On the other side of the ship, over here, we had a similar structure welded on to the ship. And this one had a water pump at the bottom. I think the next picture is a little better here. So basically down there, about 0.5 meter under the surface, we had a pump that was connected to the ship's battery and would pump water into that garden hose through this equilibrator here. In the equilibrator, the gases are driven out of solution, and then we sampled that gas through that little tubing here, and we drilled a hole in the kitchen wall to get into the kitchen or the lab into this cavity ring down spectrometer, which is a device that allows you to measure the concentration of CO2, the concentration of methane, the stable isotope composition of methane, and the stable isotope composition of CO2 all at the same time. So very, very handy. The instruments behind it are pretty crazy. You can ask Sharon about it, but basically you need to dry the gas before you can sample it. So there's a lot of pumps, a lot of gauges, a lot of drying steps before you can actually get your measurement. In addition to those two things, we had a weather station installed because you need the wind direction, the wind speed, humidity, pressure, and we also had two sensors for long wave and short wave radiation to get a handle on the heat fluxes. One of the goals in this expedition was not just to measure the concentrations, but if you want to understand the gas flux between the water and the atmosphere, you also need to have a metric that allows you to estimate the mixing, or more precisely, the gas exchange coefficient that you need in order to really calculate the fluxes. And for that, you need a lot of information on what the mixing characteristics of the surface waters are. And that's what we used, partially the ADCP, but we also had separate sensors for temperature, high frequency temperature measurements, and in the end, actually a model that allowed us to calculate the gas exchange coefficient, which you then multiply with the concentrations of the gases, and that gives you the flux of CO2 and methane either out of the water or into the water, right? And this actually worked. The question when we did this was, can we really do that? This was a proof of concept, kind of a deal. It was nerve-wracking. Don't even ask me how I got all those instruments into Russia on a 4-week time scale. That was a story for later, probably. But anyway, what you see here then is the actual concentration of CO2 in green and the stable isotope composition of CO2 in red. Some of the interesting things is, we started out with very high CO2 concentration, and that had to do with the fact that about 60 kilometers upstream, there was a reservoir, and the reservoir was drained from the bottom. And bottom waters have a lot higher CO2 concentrations, and it took about, I think, 150 kilometers or 2 days for that signal to really mix out, which is kind of an interesting side aspect. But you also see that there's regions where the CO2 concentration is actually lower than in the atmosphere, which means that something is consuming CO2 in the water, which is phytoplankton that live, in the river. But for the rest of the trip, you see a lot of sources of CO2, and I should say that we did this tuition between the end of July and the middle of August, which is outside of that very productive season. So this was kind of the easy season. If you really want to know how much CO2 and methane comes out of this, you would have to be there when the ice breaks up, which is a lot more challenging to do. We planned to do it, but then geopolitical changes made it difficult to get into Russia at the time. So we haven't done that, but the proof of concept, at least, has worked. And this basically shows you how the estimate of the gas exchange worked out. So in this case, you see two lines. The gray line would be a estimate that is only based on wind speed, which you do regularly when you determine gas exchange in the ocean. And the magenta line here is our estimate based on actual measurements of turbidity in the surface waters. And you see that for the most part, you are off by a factor of 2 to 4, using our approach versus the Wannenkopf approach, which is only based on wind speed. So turbulence in the water, obviously, it makes sense, and it is very important in river systems in order to do that. And this would then be the fluxes, so not just the concentrations, but the actual fluxes of CO2 and methane. Methane generally much lower on this scale. CO2 flux is higher, and if you would do the math, for this one summer month, you would have something about around 59 gigagrams of carbon per month in form of CO2 and about 2.4 gigagrams in form of methane. So that was really exciting for us. However, the biggest challenge that we haven't solved completely so far is that we observed a very substantial shift in the carbon isotope composition of dissolved inorganic carbon during the snowmelt period. And so that is basically what you see in this plot here, where you have the latitudes on the x-axis and the stable carbon isotope of DIC on the y-axis. The open circles here would be your snowmelt season. The triangles here are typical late summer season. And you see that there's a big shift in the isotopic composition of DIC during that time. Sometimes as much as 10 per mil, which if you are working with a large pool like DIC, if you move that much, that is a biggish number. And so the question that we are asking now is how much of the lateral flux from those watersheds are we actually missing with our current estimates? I think that those fluxes have not been included. And so even without very solid data at this point, with a back of the envelope calculation, we would estimate that this not accounted for carbon flux could be as high as 100 teragrams carbon per year for the Pan-Arctic watersheds. And that would represent about 30% of the current carbon sink that we think that those ecosystems represent. So currently we think that the boreal forests are taking up more CO2 than they release, and they release more methane than they take up. But quantitatively, the CO2 is a lot more important, right? And so if you change this by 30%, you would change that sink factor by quite a bit. And one of the questions that, climate scientists are asking is it possible that that high latitude system could actually become a source of CO2 and that it wouldn't matter if we would change fossil fuel combustion? And changing this estimate by 30% would get us much closer to this possibility. But this is sort of a little bit, reaching into the future, and a lot more work needs to be done before we can solidify that statement. So that gets me to the next story that I wanted to tell of the Central Arctic Ocean. And you see I have my conclusions already listed here. It is still necessary to have an icebreaker to get across the Arctic Ocean. It's still stunningly beautiful. And you still need to watch for those guys when you're running around on the ice. So they are still around, and most of them actually look pretty healthy still. So what were we doing here? So this is an old picture that I think was actually taken in Port Aransas, if I remember right. Some of you, not all of you, remember Ron. And Ron, one of Ron's hobbies was ultrafiltration, right? And so ultrafiltration would allow you to take 400 liters of seawater samples and compress it down to 125 milliliters. And so that is basically what we did in the Arctic then, initially. Just think about this. You're on a ship and you are asking for 200 liters of water sample. Everybody hates you, right? Because this is a lot of water. But what you see here is the difference when you concentrate different water samples. And so here you have a sample from the surface of the Greenland Sea from 1,500 meters depth, 3,200 meters depth. And if you look closely, you see how the color kind of gets less and less, right? And then on the other side, you have the Yenisei River, which is basically Coca-Cola, very concentrated. But then you have this guy in between. And this sample was collected from the East Greenland current. That's the current that flows from the Arctic Ocean to the North Atlantic. And you, with your open eye, with your naked eye, you can already tell the story, right? There's a lot more color coming from the Arctic than what you have in the Greenland Sea. And so that basically started me to think about, okay, how do I use this for my advantage, right? And we talked about developing instruments today. And when I worked at the Alfred Wegener Institute, which still had a department for instrument development, I was talking to the physical oceanographers and they said, that's interesting. Somewhere we have this instrument rolling around that a guy built to measure the Amazon river plume. And let's bring this the next time and we'll see if it actually works. And so that's what we did. And so that's when we started to really think about optics in order to describe the Arctic. And so what you see here is a multi-year data set, somewhere between 2005 and 2015, I think. You see the concentration of dissolved organic carbon, very clear, big signals coming from the Siberian rivers, and a much smaller signal coming from the Mackenzie. The picture on the right, however, are the optical properties. So you can take a sample, and one of the big advantages of optical properties, it's a cheap measurement, it's a fast measurement, and everybody can do it. And so what you see here, again, if you remember my initial plot of the transpolar drift that flows from Siberia all the way to the Atlantic, this is exactly what this plot would tell you. You got all that river signal here, and it comes across the Arctic in the transpolar drift and eventually goes back to the North Atlantic. It also tells the same story that we looked at with the sugars. The Mackenzie has a much smaller signal as well. And that pretty much agrees with an earlier study that we did using satellite data, where you see basically a lot of river DOM or optical signals along the coast. Of course, the disadvantage of remote sensing is it can't see through ice. And so you don't get that information from the central Arctic. All right, this is getting a little more boring, but what it tells you here is the vertical distribution of dissolved organic carbon, as well as the vertical distribution of colored DOM, which is measured with that instrument that the guys at the RB had laying around. And so you see that this gives you a lot more interesting information And the advantage is you can see it as the samples are collected. So you can actually use it to guide your sampling program, right? And one of the things I wanted to point out, in the Canada Basin, you have this very unique distribution of the fluorescence that's very different from all the other basins in the Arctic. And here's really the bottom line of the distribution of the colored dissolved organic matter. So this is a section throughout the entire Arctic Ocean, going from Svalbard here on the European side all the way to Alaska. And the color that you see here is that optical signal that you get from that spectrophotometer. And you see that throughout the Arctic, you have the signal in the subsurface, which is very unique. No other ocean basin has that signal. The Arctic is the only place where you find such a distribution. And you can see it in 2005, you can see it in 2007, and you can see it in 2015. So it is really a permanent feature in this basin. What else did we learn? The other thing that was striking is that the distribution of that optical signal was always along so-called isopycnals, which means it's a physical process that drives the distribution. It's not a biological process. The other thing that is important to realize, and I haven't talked about it too much, is that this signal is concentrated in the halocline. So the halocline in the Arctic is a layer that separates the warm Atlantic water that sits at about 300 meters or 500 meters. And that halocline separates that heat from the ice. So without that halocline, which basically serves as an insulation, warm water from the Atlantic would easily reach the ice and probably melt the ice within a season. So people, again, are very interested to understand how this halocline in the Arctic is maintained. However, we also realized that that optical signal that we see across the Arctic is not exclusively coming from the rivers. There is a second source for those colorful components. Now it gets really crazy. This is just to point out that you can use a combination of salinity, of oxygen isotopes of water, and nutrient ratios to distinguish different water origin. You can say this is Pacific water, this is Atlantic water, this is river water, or this is water from ice melt. And so this is something that has been used for a very long time in order to describe the oceanography in the Arctic Ocean, right? So we then started to compare our optical signal, our CDOM signal, to those parameters derived from that water mass decomposition model. And what we saw, surprisingly, that the one thing that explained the distribution of that organic matter optical signal the best was the fraction of sea ice melt. So think about it. Sea ice melt, what does it do? If you melt sea ice, it dilutes everything else, right? And so that makes sense. However, within that fraction of sea ice melt, you have a hidden piece of information, and that are those negative values. If you have negative values, that tells you that this water that you're looking at has experienced freezing or the injection of brine. So if you take a bucket of seawater and you put it in your freezer and you wait a certain time until about half the bucket is frozen and you pull that ice plug out, then you will see that all the fresh water is in the ice, but all the organics that were in the seawater are still in solution. So freezing seawater separates the water molecule from everything that's dissolved in it. definitely everything organic. So that was something that, we figured out a while ago, but we never saw it on a large scale like in the Arctic Ocean like that. And so this plot that looks familiar to the last one, which is true for the color, but the black lines that you see here are now the fraction of sea ice melt. And you see again that the distribution of that optical signal follows the line of that sea ice brine formation very well. So now we know that the most important factor for the distribution of that organic matter in the Arctic is the physical process of sea ice formation and melt, most likely on the shelves. The next problem that we had with this comparison of the optical characteristics and the different types of water masses was that in the Canada Basin, surface waters, we never saw any signal for that CDOM or for that organic component. But if you look at the fraction of meteoric water, which stands for river water for all practical purposes, this would tell you there's a lot of river water in the surface of the Canada Basin. But there was no organics in there that would tell the same story. So now we have a problem that those two indicators don't match up, right? And so what we did then is to look at some of the information by separating the data set. So we have the green data, which is the Eurasian basin, and the blue data, which is the Canadian basin. And then you look at the concentration of that CDOM or that optical signal from the organics against the amount of river water or meteoric water in those samples. And what you see is that in the Eurasian basin, it makes sense. The more river water you have, the higher your optical signal. But in the Canada Basin, it would tell you the opposite. The more meteoric water you have, the less signal from the optical properties you get. And then we said, okay, let's get away from this confusing metric here and just look at salinity and the stable oxygen isotope concentration of the water molecules, two relatively conservative metrics, which is something you really like as an oceanographer because you can, rely on it. And again, we split the data set between the Canada Basin and the Eurasian Basin, and you get two very different relationships. If you look at that equation here, it sort of allows you to theoretically estimate the value that you would find at zero salinity. And it would indicate that in the Canada Basin, you are dealing with a deficiency of salinity, of salt, which means that if you think about ice formation, that the Eurasian Basin seems to be the place where you form more sea ice than you melt, while in the Canada Basin, you seem to melt more sea ice than you form. And that's why those two basins look so different. And so what we did, don't worry about all those numbers, we basically replaced one of those equations that used to be the nutrient relationship with our CDOM information. And then we calculated the different fractions, and what I show you here is the amount of ice melt. I was always bothered by the classical way because it always predicted very low levels of ice melt. And I was always skeptical about that. And so when you look at this, on the bottom panel, you see the classical approach of calculating the sea ice melt. And on the top, you see the modified approach using the CDOM signal along with salinity and oxygen isotopes. And you see here, there's a lot more ice melt according to the new model than you have in the classical model. However, there's also an interesting pattern in this one because it would indicate that in that transpolar drift, right, where you usually have this massive signal of sedum, that most of that water comes from brine. And that, of course, would make sense if the Eurasian basin is dominated by sea ice formation rather than melting. And that signal is carried in that water mass that comes across the Arctic. So we are still very interested what drives the halocline and where that halocline signal comes from. And so what you see here is basically another oceanographic tracer that allows you to distinguish Pacific water from Atlantic water. And the profile that you see here is basically the sedum distribution in the Arctic Ocean. And you see that on the top side, all the water comes from the Pacific. On the bottom side of that peak, it seems to come from the Eurasian side. So both of those water masses contribute to that sedum signal. And while on the Eurasian side, the dominant source for sedum is the rivers, on the Canadian side, the dominant source of that sedum seems to be the sediments. The Chukchi Sea, you can ask Ken here, who knows all about it, is a very productive area, right? It's also relatively shallow, so a substantial fraction of the primary production makes it to the sediment surface. It gets degraded on the sediment, and when it is degraded, it seems to release a component that has very strong optical characteristics that we pick up with that sedum fluorometer in the open Arctic. And so think about the Arctic as being mostly melted along the shelf seas, where you have also a lot of fresh water coming in. And so when you refreeze those shelf waters, you reject all the organics. Those organics then sink towards the bottom because they are slightly lighter, slightly heavier. And then they run off from the shelves into the central Arctic. But depending on where you are, it will give you a different signal, either a river signal or a sediment signal. And so where are we going next? So those are plans for the future. You see, I already did a trip last summer and collected samples for measuring lignin phenols. Those are our best descriptor for terrestrial organic matter. I just had a meeting this morning with people that are going out in the Arctic in this summer, and they will, or at least they're planning to collect, samples at those stations. And so I am getting additional samples for that, which gives me a pretty good coverage within a couple of years. And then hopefully next year, we are able to cross the entire Arctic Ocean again using the Healy US icebreaker. And then we would have a very fantastic higher resolution data set on lignin phenols. And the plan is to use those lignin data for those water mass decomposition models in order to better describe the sources of fresh water in the Arctic Ocean. Okay, then you might ask a question, how are we doing with time? A little bit. What happens to all that organic matter that you find in the Arctic Ocean? So this is some work that I did a very long time ago. Here's Greenland. So basically, this is the gateway where waters from the Arctic flow in that East Greenland current towards the North Atlantic. And you can see, again, this is that sedum signal that we were talking about all the time. You clearly see it at 79 degrees north. You clearly see it at 75 degrees north. You see it at 71 degrees north. And even in the Denmark Strait, you can still see that Arctic river signal going into the Atlantic Ocean. Some chemical analysis that we did even found terrestrial organic matter in the deepest branch of the North Atlantic deep water. So that's another mind-blowing fact, right? That you can take river water, which is warm and has no salinity, zero salinity, and you transform it so much that it ends up in the Denmark Strait overflow that then enters the Atlantic Ocean and goes south. And that gets me to the final chapter of this story, and thinking about how the Arctic is connected to the lower latitudes, and in this case, specifically to the Gulf of Mexico. The Gulf of Mexico deep water in here is actually ventilated by upper North Atlantic deep water. So water that originates in the northern high latitudes. And this program, which this lady here is responsible for partially, had a lot of data. You know, they were measuring the volume transport through Yucatan Strait for many, many years, and with moorings. And you see the mooring line here in Yucatan Strait and another mooring line in the Florida Strait. One problem with the Gulf of Mexico is that there's not a lot of oceanographic data, right? So we found 2 radiocarbon data or profiles. That's it. That were taken in 78 and I think in 93. That was all. So when we started to design part of that CGOM program, we said we should take another set of high quality radiocarbon samples. And that's basically what we did. And it allowed us to come up with some very interesting timelines. This is some work done on the deep water temperatures in the Gulf. And you see data that were collected from moorings at 3,500 meters. Just think about it, 3,500 meters deep. And you see over the time of between 2003 and 2019, you see a very nice trend of increasing temperatures. It's not a whole lot, but it's a consistent trend that has been going on for a long time. The same in old CTD data. Every consecutive year that this program collected CTD data, the temperature was higher, very consistently. And so discussing this with the group at CCS, in the beginning we were skeptical. Is this real? Are our sensors drifting? What is going on? But after a while, also when we looked at the radiocarbon data, there seems to be a shift that's happening in the Gulf of Mexico. And so what we decided to do is trying to make a connection to the North Atlantic. So we were estimating how long it would take to get the North Atlantic deep water into the Caribbean and then flush the Gulf of Mexico, right? And so we estimated it takes about 15 years to get to the gateways of the Caribbean here. It takes about 45 years to ventilate the deep Cayman and Yucatan Basin. And then it takes another 100 years to ventilate the entire Gulf deep water. And so that would take you to a time scale of 160 years. So it takes about 160 years to start the ventilation in the North Atlantic and then flush out the entire Gulf one time, right? And so if those changes that we observed in temperature and in the radiocarbon data, that would mean that these changes would have to be initiated 150 years ago, something like that. Don't pin me down, but on that time scale. And that would mean that it would put it right at the end of the Little Ice Age, which was from the medieval times to sometime 1880, something like that. And that would be consistent with a lot of the modeling studies and some of the paleoceanography studies that actually claimed that something must have shifted in the North Atlantic around that time when the Little Ice Age ended. And so in the end, I wanted to put something out there that is a little controversial, but it's a very intriguing theory. I don't know how many of you have heard of Rudiman, but he has the hypothesis that human activities have changed the climate on this planet much sooner than we think. And so his argument was, when he looks at the orbital forcing, so the influence of basically the distance of our planet from the sun and the way it's oriented towards the sun, we should have been in a cooling phase for at least 5,000 years, right? You see that amount of energy coming in from the sun? It's clearly declining here based on the orbital force. And he argued that the beginning of the Little Ice Age was actually that initial entering of that cooling phase. And then he said, this cooling phase couldn't continue because human activity already put so much more greenhouse gases in the atmosphere by early agriculture, deforestation, that it actually kneecapped the orbital forcing and brought the climate back up, which otherwise we would have been a lot cooler right now. And that is basically what he shows in this plot out of one of his papers. And you see that we are way overdue for in terms of the summer insolation. We also should have a lot more ice than we actually have. This scale here is switched around, so be careful. This is actually more ice, this is less ice. And he said that the reasons for that are the concentrations of methane and CO2 that have actually, counterbalanced that initial cooling. And it's just a very intriguing theory to think about, that human activity could have stopped the orbital forcing early on. And so major conclusions then from this long journey here, I would say that there might be an unaccounted lateral flux of carbon from the vast Arctic watersheds that we have not appropriately accounted for. That we have some unresolved issues with Arctic Ocean freshwater sources that we need to revisit, as well as the transport. And we think we have a powerful tool in order to determine the stability of the halocline and also the maintenance of the halocline with those new tracers. I would say that in both of those environments, the Arctic watersheds as well as the Arctic Ocean, we have learned a lot. I would argue that maybe we know more about the Arctic Ocean than we do about the Gulf of Mexico. If you look at the number of cruises and projects, but we still have a lot to learn. There's a lot of things we don't understand appropriately to judge the changes, right? So it's like hitting a moving target, right? You're trying to see what the changes do to the system, but you don't really understand the fundamental workings of it. It's very, very tricky. Models are also struggling a lot because the Arctic is quite unique in relative to other ocean basins. And so there needs to be more exchange between observers and the people that try to simulate the changes in the Arctic. And one thing that is clear is that the ocean has a long memory. And if it turns out that we have charged the ocean with heat and greenhouse gases for thousands of years, it will take a very long time for that system to recover. And so that is just some thoughts that you might want to chew on. And with that, I say thank you for having me. I enjoyed being here. And thank you to all the people that helped me and contributed to those stories along the way. The people in Russia that really helped me out. It wouldn't be possible without them. My colleague Carl at Tamuk, responsible for a lot of the development of the new methods. and the folks at CISESA, which now Sharon is not part anymore, but she was part of that group in Baja California. Funding from the National Science Foundation, Texas A&M, the Alfred Wegener Institute, the Swedish Secretariat for Polar Research, and all the students, undergraduate students and graduate students that have contributed to that along the way. Thank you.